All right, it's my great pleasure to introduce today's speaker, Dr. Ying Yang. She's an assistant professor in the Department of Molecular Pharmacology and Physiology at University of South Florida in Tampa, Florida. And the main research interest in Dr. Yang's lab is to understand the biological functions of the transcription factor FOXO1 during lymphatic development and lymphatic disease, and to develop novel therapeutic targets for lymphedema. Her NIH-funded lab is the first to stimulate the growth of new lymphatic valves in a physiological manner by deleting FOXO1. And this has the potential to treat several types of primary lymphedema where valves regress or disintegrate. Dr. Yang has received numerous awards, and one that I'll mention here is the 2021 NAVBO Springer Junior Investigator Award. So the title of her presentation is A Novel Mechanism to Enhance Lymphatic Valve Formation, Therapeutic Implications for Lymphedema. Thank you, Ying.
Yeah, thank Michael and LGDA for this opportunity to share our work on the novel mechanisms that regulate the formation of this specialized structure in the lymphatic vessels, the lymphatic valves. Uh-oh, why it doesn't listen to me? Okay. So the lymphatic vasculature is a network of lymphatic vessels that are connected by lymph nodes. It is the main transportation pathway of tissue fluids to maintain fluid homeostasis. It also transports immune cells for immune response. And the lymphatic vessels in the gut villi absorb dietary fats, which is essential for lipid absorption. Dysfunction of the lymphatic vasculature is associated with many diseases. Most directly is lymphatic diseases, which includes lymphedema and lymphatic malformations. In most recent years, studies have shown that lymphatics plays very important roles in many other diseases, including hypertension, Alzheimer's disease, atherosclerosis, myocardial infarction, aging, and metabolic diseases. And studies have shown that enhancing the function of the lymphatic vasculature alleviate the severity of all these diseases. To execute these important functions, lymphatic capillaries absorb interstitial fluid, macromolecules, and cells to form lymph through their highly permeable junctions. Then the downstream connecting lymphatic vessels transport lymph, which will be returned to the blood vasculature through the valves between the thoracic duct and the subclavian vein. One of the unique features of the collecting lymphatic vessels is that they have valves. These intraluminal bicuspid structures are the gatekeepers to maintain the unidirectional lymph flow against a hydrostatic pressure gradient. As you can see in the video, the valve leaflets open and close in response to the upstream and downstream pressure of valves to ensure that lymph can pass but cannot go back. Defective valves cannot close tightly to prevent retrograde lymph flow, which can lead to lymphatic diseases such as lymphedema. There are two types of lymphedema. Primary lymphedema is also called congenital lymphedema, which is caused by inherited mutations in genes that regulate lymphatic development. This is an image from a patient with lymphedema distichiasis, which is caused by mutations in FOXC2 gene. And FOXC2 is a transcription factor that is crucial for the formation of lymphatic valves. Lymphoscintigraphy is used to visualize the function of the lymphatic vasculature. As you can see here, the radioactive tracer was drained by the lymphatic vessels to the lymph nodes in healthy patients, while tracer was absorbed but could not be efficiently transported due to valve failure. The presence of the dermal backflow indicates that lymphatic valves are dysfunctional in lymphedema distichiasis patients. In addition to FOXC2, there are many other gene mutations that cause congenital lymphedema, have valve defects in their corresponding knockout mouse models, as shown in this list, which indicate that valve defect is a causative factor for congenital lymphedema. Secondary lymphedema is also called acquired lymphedema, which is caused by injury to the lymphatic vasculature after birth. The most common case of secondary lymphedema in developed countries is from lymph node removal surgery during cancer treatment. Breast cancer patients are the largest population that have secondary lymphedema. 20 to 50% of them develop secondary lymphedema after surgery. And similar to the congenital lymphedema patients, secondary lymphedema patients also show dermal backflow in lymphoscintigraphy, which indicates that these patients also have dysfunctional valves. So how do valves form? Work from Tatiana Petrova, Mark Kong, and Taya Makining's lab has shown that oscillatory shear stress from oscillatory lymph flow is critical for the specification of valve cells. 3 transcription factors, PROX1, FOXC2, and GATA2, are upregulated in response to oscillatory shear stress, which indicate that the mechanotransduction signaling pathway is essential for lymphatic valve to form. In the mechanotransduction signaling pathway in blood endothelial cells, AKT is a major downstream effector of after shear stress is sensed by the molecules on the cell surface. Similar to blood endothelial cells, our previous work showed that in cultured human dermal lymphatic endothelial cells, phospho-AKT, the active form of AKT, was upregulated in the presence of oscillatory flow. So our question was, what is the downstream target of AKT in the mechanotransduction signaling pathway that regulates the formation of lymphatic valves? So after we searched literature, we found out that it is well documented that in many cell types, the nuclear activity of a transcription factor FOXO1, foxhead box protein 1, is regulated by AKT. As a transcription factor, FOXO1 protein locates in the nucleus and binds to DNA to regulate gene expression. Upon stimulation, phospho-AKT deactivates FOXO1 in the nucleus by phosphorylation, which excludes FOXO1 out of the nucleus. However, the function of FOXO1 in the lymphatics was unknown. So to study this, we deleted FOXO1 from lymphatic endothelial cells using the PROX1 CRIA T2 mouse model in embryos, postnatal pups, and adult mice. And we also used PROX1 GAP reporter mouse line to visualize the lymphatic vasculature. Since the embryonic and adult data are consistent with the postnatal data, I will only show the data from the postnatal deletion here. And as you can see, compared to the control vessels, FOXO1 deletion induced new valves to grow. These valve-forming areas were rarely seen in the control vessels. Our quantification data confirmed that valve number per millimeter in pups with FOXO1 deletion was significantly higher compared to the control at 4 different time points. And meanwhile, deletion of FOXO1 did not affect vessel diameter or branch number. New valves were not only induced in the mesentery vessels, but also in the axillary vessels. Therefore, FOXO1 is a repressor for lymphatic valve formation. And to further characterize these new valves that were induced by FOXO1 deletion, we performed the whole mount immunostaining and a confocal imaging to investigate whether these newly formed valves expressed valve-forming genes, such as PROX1, FOXC2, GATA2, KL4, and integrin alpha 9 as the control valves. Our results show that the new valves express all these valve-forming genes, which indicates that these valves were in the process of normal development. And moreover, these valves did not display any defects in junction protein expression. And these junction proteins are important for maintaining the integrity of vessels and the valves. So these results indicate that the new valves induced by deletion of FOXO1 appear to have normal expression of valve genes. And besides FOXO1, another FOXO family member, FOXO3, has been shown to have important function in blood endothelial cells. So we use the same PROX1 CRE-ERT2 mouse model to delete FOXO3 in the lymphatic vasculature. Deletion of FOXO3 did not induce new valves to grow. There was no difference in the valve number between the FOXO3 knockout and the control mice. We also generated a double knockout of FOXO1 and FOXO3 to check whether FOXO3 activity is required for FOXO1 deletion to induce new valve to form. In a double knockout mesentery, we still observed many valve-forming areas. The number of valves was significantly increased in the double knockout compared to the controls. And this valve number was similar to FOXO1 single knockout. So these data showed that FOXO1 deletion in lymphatic endothelial cells induce new valves to form, and this activity is independently of FOXO3. So why is growing new valves important? Since adding new valves to a healthy vessel is not necessary. Because we think FOXO1 deletion might be able to rescue lymphatic valve defects in lymphedema disease. So as I mentioned earlier, FOXC2 heterozygous mutations cause lymphedema distichiasis syndrome. And since FOXC2 heterozygous mice is an animal model for lymphedema distichiasis, we deleted FOXO1 in the FOXC2 heterozygous mice to test the rescue. To delete FOXO1 in the FOXC2 heterozygous background, we used the FOXC2 CRE-ERT2 mouse model, in which one allele of FOXC2 was replaced with CRE-ERT2. Therefore, this mouse model is both heterozygosity for FOXC2 and has CRE-ERT2 activity. We induced the deletion of FOXO1 right after birth and analyzed the lymphatic vasculature 3 weeks later. Our data showed that FOXC2 heterozygous mice had about 50% fewer valves at postnatal day 21 compared to the control. And deletion of FOXO1 completely rescued the valve number to the wild type level. And also additionally, FOXC2 heterozygous caused a significant reduction in the number of branches, and deletion of FOXO1 rescued the branch number as well. To further confirm that the morphology of valves and the expression of valve genes were restored in the FOXC2 FOXO1 rescue animals, we performed confocal imaging of PROX1 FOXC2 V-cadherin and integrin for alpha 9 immunostaining. We showed that although the defective valves in the FOXC2 heterozygous animals express PROX1 and FOXC2, they did not have the valve leaflets shown by integrin for alpha 9 staining. However, the loss of valve leaflets was restored by deletion of FOXO1 shown with integrin for alpha 9 staining and the quantification. More importantly, not only the valve number and the morphology, but also the function of the valves was restored to the wild-type level with FOXO1 deletion. To test the valve function, a single vessel was cannulated on 2 glass pipettes. Output pressure was slowly increased while input pressure remained constant. Valve leaflets from healthy vessels close tightly together to prevent back leak of flow, and any back leak can be quantified by the servo node pipette behind the valve leaflets. So here we show that all leaky valves in FOXC2 heterozygous mice were restored to be able to close tightly against the 2 different pressure gradients to prevent backflow of fluid when we delete FOXO1. So what is the mechanism of how FOXO1 deletion lead to new valves to form? Our knockdown and flow experiments show that FOXO1 represses the expression of valve-forming and flow response genes, including FOXC2, GATA2, KL4, KL2, and with and without flow treatment. This is from mRNA level, and this is from protein levels. And the flow experiment also showed that the nuclear localization of FOXO1 was severely compromised in the presence of oscillatory flow, which is in line with the upregulation of phospho-AKT and phospho-FOXO1 under the same flow condition. Our chromatin immunoprecipitation assay showed that FOXO1 binds to the promoter area of FOXC2 to repress its expression. Therefore, our study revealed that in the mechanotransduction signaling pathway in response to oscillatory shear stress, AKT activity goes up, and the nuclear activity of FOXO1 goes down. Which allow the upregulation of valve-forming gene expression. So this whole process facilitates the formation of lymphatic valves. In addition to genetic methods, we tested the effect of a commercially available FOXO1 inhibitor in cultured human dermal lymphatic endothelial cells. As you can see, the FOXO1 inhibitor significantly increased the expression of valve-forming gene and flow response genes on both the mRNA level and the protein levels. And in addition to the cell culture data, in vivo injection of the FOXO1 inhibitor significantly increased the increased valve formation in FOXC2 heterozygous mice by showing a partial rescue of the valve number. So after we gained some insights into the downstream of AKT in the mechanotransduction signaling pathway, we wanted to understand a bit more about the upstream of AKT in the mechanotransduction signaling pathway in response to shear stress. In many cell types, Richter in the mTOR-C2 complex activates AKT by phosphorylation. However, the role of Richter in the mechanotransduction signaling pathway remained unclear. So we deleted Richter from lymphatic endothelial cells using the same PROX1 CREAT2 mouse model in embryos and postnatal pups. As you can see, both stages showed a significant loss of valves upon Richter deletion. And this valve loss phenotype was observed not only in the mesenteric lymphatic vessels, but also in the ear, skin, diaphragm, and axillary vessels. Mechanistically, we found that Richter is necessary for the upregulation of phospho-AKT, as well as valve-forming and flow response genes in response to oscillatory flow. And consistent with the downregulation of phospho-AKT and the Richter knockdown cells, our whole mount immunostaining and the quantification results show that FOXO1 was retained in the nuclei of the Richter knockout cells in vivo. And our FOXO1 nuclear overexpression mouse model further confirmed that nuclear FOXO1 is a strong repressor for lymphatic valve formation. Therefore, we tested the hypothesis that by deletion of FOXO1, we can rescue the valve loss in Richter knockout mouse. Here is the result. As you can see, we successfully completely rescue the valve number in the Richter knockout by deletion of FOXO1 to the wild type level. So in summary, our studies demonstrated a novel role of Richter, AKT, FOXO1 signaling access in the mechanotransduction signaling pathway during lymphatic valve formation. So in response to oscillatory shear stress, Richter activity is upregulated to increase the activity of AKT by phosphorylation, which exclude FOXO1 out of the nucleus to facilitate valve formation. In the loss of Richter, AKT activity goes down and the FOXO1 nuclear activity goes up, which represses valve formation. So our study, the implications from our study is that we believe the ability to stimulate the growth of lymphatic valves de novo in a physiological manner provides opportunities to find potential targets for lymphatic diseases where valves are disintegrating. And lastly, I would like to thank all the members from my lab, especially Lutz and Richard, as well as Oladi, who worked extensively on these projects. I would also like to thank all my collaborators for their tremendous support, particularly Michael Davis and Jorge for performing the valve function tests, Taya McLean, Yang Hong, and Satish for sharing their mouse models. And I cannot complete these projects without the support from our department and the imaging core facility and mouse facility. And last, I would like to thank NIH for funding support. Thank you everyone for your attention, and I'm happy to take questions.
Great presentation. Really exciting work. I'll start the discussion, and if people have a question, they can just raise their hand and then unmute themselves and ask. I was curious if any of the models have lymphedema or chylous reflux, and whether loss of FOXO1 can reduce those effects.
Yeah, it's a great question. So as you know, we all work on, we both work on mouse models. And for mice, based on their size, it is almost impossible for the mice to develop any lymphedema as human, because the mouse they do not experience the same the gravity level as human because of the size and of also the activity, the more activity they have. So in order for mice to get edema, in our previous experience is that most of the lymphatic vessels have to be abolished completely in order for them to develop lymphedema. So although 50% of loss of valves can cause lymphedema distichiasis in human patients, but FOXC2 heterozygous, for example, these mice do not develop lymphedema, like edema phenotype as human patients. And the chylothorax. So in terms of chylothorax, based on our other experiments, as well as the studies from Donald McDonough's lab, suggested that valve function or the valve numbers need to be severely reduced in order to let the carothorax to be happen. So in that sense, FOXC2 heterozygous mice do not have carothorax.
All right, thank you.
And also believe like if we can use FOXO1 to restore valves in the mouse model that develop carothorax, and we believe we can rescue the phenotype.
Elisa?
Yeah, hi. This was hi, Ying. This was beautiful, very fascinating work. I was very curious about what is, how is the shear stress getting translated into Richter signaling? Do you have you looked into that?
Actually, I have not looked into that. So far, I just know in response to shear stress, Richter level is going up. But I do not know the signal between the shear stress, for example, PCAM, V-cadherin, or even VEGF receptors, all those, when they are activated, sense the shear stress, then how that increase the Richter expression level. So it's a really good question. We don't know.
Well, looking forward to hear about that. I'm sure you're into it.
Thank you.
Thank you.
Mike, I'm technically challenged. I can't figure out how to raise my hand. So maybe I can, since I see that nobody else has raised their hand, maybe I could jump in with my question.
Certainly.
Nice talk, Ying. The MAP kinase pathway, which I have a very strong interest in, we have determined is also a critical regulator of valvulogenesis, in the sense that any type of activation of that pathway during that process is bad news for valvulogenesis. So if we block any MAP kinase activation during valve development, you also see a large increase in the number of valves. So with that in mind, we know that AKT crosstalks with the MAP kinase pathway. It phosphorylates MAP kinases and inactivates them, such that if AKT is not being activated properly, you'll have dysregulated MAP kinase activation. I was wondering which would also impede valve development. So I was wondering, therefore, if in your Richter deficient mice, where there's defective activation of AKT, whether that is 100% explained by aberrations in FOXO1 exit from the nucleus, or there's also room for the notion that dysregulated MAP kinase activation contributes to that phenotype. And if you have tried to rescue the Richter phenotype with MAP kinase inhibitors.
I see. It's a really interesting question. Yeah, so I totally agree with you that, so from our experience, also your work, that AKT activity, like increase the AKT activity, is very important for valve to form. Meanwhile, the inhibition of ERK activity is actually also beneficial for valve to form. So in our Richter knockout mouse model, we actually have not checked the ERK signaling activity. So in our hand, we checked the AKT activity, which showed here. So this is actually the older version. We update this version. But as you can see, this is the serine 470, 437 site of AKT, which is downregulated when we knock down Richter. And but also you can see the activity of AKT is not completely abolished when we knock down Richter, and which is actually quite consistent with our in vivo phenotype is that we looked at the phenotype beyond 3 weeks of Richter deletion, and these mice just maintained low level of valves, and not all the valves are going away. So, so I do not know in this case whether you know, the residue of AKT as well as the ERK signaling pathway have, you know, function on this in this phenotype. So it will be good to check the level of ERK signaling in these mouse.
Right, not so easily done in vivo, unfortunately, but
right. I can do it in vitro, yeah.
But you could also just administer a MAP kinase pathway inhibitor to the Richter deficient mice to see if that can rescue any form of the phenotype.
Mm-hmm. And in our hands, at least like in vivo, when we delete FOXO1, and we deleted both one allele and two alleles of FOXO1. As you can see, FOXO1 in our hands, we also know that FOXO1 deletion is FOXO1 is a dose-dependent transcription factor as well. Is that you have a non-significant rescue of valve number when we delete one allele, but sorry, my mouse is so touchy. And when we delete both allele of FOXO1, we bring the number back to the wild type level completely. So I do not know when we delete FOXO1 whether that has, you know, anything like affected the ERK pathway as well.
Yeah.
That part we don't know. It would be good to check that. Thank you for the suggestion.
All right, we have a question from the chat that I'll read. This is from Brian. What else is FOXO1 responsible for? Is inhibition in humans feasible, or do you anticipate significant side effects?
Yeah, so FOXO1 is like many other transcription factors that it is expressed in multiple cell types. So particularly during development, FOXO1 has very important and versatile functions in many tissue types. So when we did an embryonic deletion, or when we delete FOXO1 during the development of lymphatic vasculature, in addition to inducing lymphatic valve to form, we also observed the increased proliferation of lymphatic endothelial cells, which is consistent with the result from deletion of FOXO1 in blood endothelial cells. So that leads to the increased diameter of lymphatic vessels when we delete FOXO1 in the embryos. So, and also systematic inhibit FOXO1 during development is gonna cause other issues besides in the lymphatic vasculature. So FOXO1 is also very important for T cells development as well as others. So I do not anticipate that during development to inhibit FOXO1 systematically. However, postnatal deletion or later deletion that we did in the adults appear to have much less side effect in terms of like inducing the diameter of vessel, like increase the proliferation of lymphatic endothelial cells. So both postnatal and adult deletion of FOXO1 appear to have very specific effect that the valve number is increased. But still, FOXO1 in the adult it's still expressed in many other cell types. But so far from a literature, from my reading, is that in the adulthood, FOXO1 inhibition appear to have more beneficial effects than earlier deletion. For example, if you inhibit FOXO1 is a potential target for diabetes, because FOXO1 is important for regulating glucose levels. And a lot of study have been done to analyze inhibition of FOXO1 in the diabetes setting that can alleviate the insulin resistance and phenotype. And so my anticipation, I think, it would be still want to pursue cell type specific inhibition of FOXO1 if we can.
And I was just thinking too, with sort of a pharmacological approach of blocking this,
Yeah.
do you think that you would have to be maintained on the drug, or do you think that these effects would be durable? So you give the inhibitor and then withdraw it and the valves would stay. Or do you think that if you take away the inhibitor, the valves would?
Yeah. So from studies so far that have been done by the groups, so the maintenance of valves also require oscillatory shear stress signaling and all that. So I would say once the valves are being induced and to form, then as long as the signal from lymph flow is being maintained in a good level, then the valve leaflets would not disintegrate in that case. But just a negative note on this, my thought is because the congenital lymphedema patients already carrying gene mutations, so they will always have that mutation. So for them, I think you probably need a constant inhibition of FOXO1 to maintain the proper level of valve gene expression. Yeah.
Well, I want to thank you once again for an excellent presentation.
Thank you.
Thank you everyone for attending and for this great discussion. And we look forward to seeing you all next month. Bye, everyone.